Bienvenue à tout le monde. Okay. Bienvenue à tous, à toutes. Mon nom est Yves Joannette et je suis ici à titre de responsable du comité de gestion du fonds Yves et Justine Sergent à l'Université de Montréal. Et c'est avec grand plaisir que je vous accueille aujourd'hui, ici dans la salle et vous en ligne, pour la remise du prix 2019, du prix Justine Sergent. J'aimerais donc simplement introduire un tout petit peu le contexte de la remise du prix, puis avec la présence du Dr Christian Baron, nous allons remettre le prix à notre lauréate de cette année. Et après, nous aurons l'occasion d'écouter la lauréate. Mais pour mon introduction, I will shift to English so that everyone will be part of the introduction and so on. So the Justine and Yves Sergent Fund was endowed at the Université de Montréal in 1998, and the first prize was offered in 1999, so it's the 20th year this year of the award. By the family of Sergent, Yves Sergent, and as well by Mrs. Edith Laubier. Madame Sergent unfortunately passed away, but she was a regular of this event up until 10 years ago. And Mrs. Laubier as well, unfortunately passed away, but she was a great defender, not only of areas such as neuroscience, but also ecology, parks, and lots of artistic group in Montreal that were supported by the foundation, the Edith and John Laubier Foundation. And Mrs. Laubier was recognized having a honoris causa doctorate from the Université de Montréal in 2009, and also is remembered in the EGLB, Edith and John Laubier Foundation, which is still active for young researchers. The Fonds Justine and Yves Sergent was created to the memory of Justine and Yves Sergent, who both passed away together on April 12th, 1994, in a complicated context of a long-standing and profound misunderstanding, including allegation of ethical misconduct, for which an inquiry was suspended 3 years after her death, without having raised any evidence of fraud. And in that sense, just to remind you, Justine Sergent was a young elementary school teacher in Lebanon in a small village who at one point met Yves Sergent, who was on military cooperation from France in this little village in Lebanon, and they really fall in love, and they married in Paris, and then Yves Sergent was hired as a school psychologist in the Protestant English School Board of Montreal, and so Justine Sergent started as a elementary school teacher in Montreal at the English School Board. But rapidly, she started psychology. She did her bachelor. She did her master. At one point, the brother of Yves Sergent had a brain tumor, and so she was attracted by the brain. And she did her PhD, her postdoc at McGill, and then she became a scholar and a scientist of the CIHR, then called the Medical Research Council at McGill. And the whole contribution of Justine Sergent to science was made within about 10 years, which is quite amazing, since she did profound contribution to the field of cognitive neuroscience. The concept of interhemispheric cooperation was certainly one of the things she introduced, but also she did seminal contribution, and I see Isabelle Peretz, Dr. Peretz, who was one of the awardees some years ago, but we're in the music area, and she did certainly pioneer imaging studies in the area of music. And so you have here some of the papers in different journals that really marked the field. And again, for a elementary school teacher starting in the English School Board in Montreal and going to this in tenure was really something really amazing. And this is the last sentence I always share each year that she wrote just before passing away with her husband. Anyways, the Justine and Yves Sergent Award honors each year a woman researcher because the family decided to turn this sad story into something positive. And given the excellence of Justine Sergent and the fact that she was really, with her husband, supported, this fund was created so that a woman, cognitive neuroscientist, would be honored, someone who had impact on the field. And so we have a management committee, Dr. Patrick Vinet, Dr. Pierre Reville here as part of the management committee, myself. And we have a scientific committee, so there's no way you can apply to that fund. It's the scientific committee who looks at the global area and identifies top people that really contributed to the field in the area of cognitive neuroscience. Dr. Daniel Bob, Dr. Rita Ari from Finland, Dr. Leslie Unger-Leider from Bethesda are all on that committee. And in the past, many people such as Kathy Price, Isabelle Peretz, was your year, Isabelle in 2009, 10 years ago. Boy, goes fast, right? Patricia Reuter-Lorenz, Ghislaine Dehaene, Rita Salmelin, for instance. And last year, we welcomed Elizabeth Kensinger from Boston. And this year, well, I'm really, really happy and proud that Professor Dr. Angela Friederici was selected by the scientific committee as the annual awardee. Angela was a pioneer in many, many ways, having a PhD in linguistics and psychology, a postdoc at MIT. Please correct me if I read wrongly your CV. She had a number of contribution to the field as a research associate at the Max Planck Institute in Nijmegen in the Netherlands, professor of cognitive science in Berlin. But certainly, Angela has to be stressed her contribution as the founding director of the Max Planck Institute for Human Cognitive and Brain Science in Leipzig, Germany. And this is really something that is amazing, just if only because she was invited to be the founding director in not an easy context, because this was just after the reunification of Germany, and when East and West were trying, well, the West was trying to support the East, so to say, to develop science. So really, chapeau. And this year we're celebrating not only the 20th anniversary of the prize, but also the 50th anniversary of the wall down in Berlin. She's a honorary professor at the University of Leipzig, Potsdam, and Berlin. She had numerous awards, and now it will show that my German is limited. The Eisenberg Fellowship and the Gottfried Wilhelm Leibniz Prize from the German Research Foundation, the Alfred Krupp Award, the Johannes Gutenberg Endowed of Professorship, and she received a doctor honoris causa from the Université de Mons in Belgique. As well as she's a member of many academies, including the Berlin-Brandenburg German Leopoldina and Academia, and the Academia Europea in Germany. The last line gives some information about her scientific contribution and production, but I will stop here and certainly just mention that it could not be more opportun to have Professor Friederici this year to celebrate together the 20th anniversary of this prize. So before asking Professor Friederici to share with us the annual lecture, I will welcome Dr. Christian Baron from the Faculty of Medicine on behalf of the Dean, who will say some words, and then together we will remit the prize, and then we'll hear the talk.
Alors, cher Yves, monsieur le directeur du consortium sanitaire numérique et ancien directeur de l'Institut de vieillissement de l'IRC, je pense qu'on peut encore le dire, chers collègues, chers étudiants et invités, merci beaucoup pour l'invitation. Je suis très content d'être ici avec vous à l'occasion du 20e anniversaire du prix Justine et Yves SERGENT, un prix de renom dédié à la reconnaissance de l'excellence et de l'impact des travaux des femmes en neurosciences cognitives. Cette année, ce prix important est remis à Dr. Angela Friederici, qui est la directrice de l'Institut Max Planck pour sciences cognitives du cerveau humain à Leipzig en Allemagne et aussi VP de la société Max Planck. Vice President. I'm very happy to be here today to represent the direction of the Faculty of Medicine, and the Dean is sorry that she couldn't make time in her agenda as she usually is happy to do, to transmit my best wishes to Dr. Friederici for the very impressive body of her work in the field of cognitive neurosciences. For those who do not know much about the German scientific system, the directorship of a Max Planck Institute is a very high distinction, reserved only for those who made an exceptional number of contributions and whose work holds great promise. The resources available to a Max Planck director are exceptional, and they may be comparable to Howard Hughes award in the United States, and even better because it's a lifetime award and distinction, and this enables them to have an even greater impact on the development of science and technology. The title of her presentation, Language in Our Brains, speaks to a question of basic importance that reflects the research interests of many of our colleagues here at the EUGM, notably on the acquisition of multiple languages, for which Montreal is a very rich ecosystem of bi, tri, and even multilingual persons, of which we have plenty here. These areas of research also interest many other colleagues in the faculties of medicine, arts and sciences, and others, and University of Montreal. Brain science in general is a big priority here in Montreal in our research community, and there are a multitude of collaborations and interactions between colleagues at the University of Montreal and at McGill. Welcome also our colleagues from the NMI. We have seen multiple important investments from the federal and the provincial government that are quite naturally also to the link to the field of data sciences, which is another one of our priorities. C'est bien naturel que le décernement de ce prix a lieu ici à l'UGM, qui joue un rôle fédérateur et unique dans le développement de la recherche, dans la formation d'étudiants et dans la valorisation des connaissances en neurosciences cognitives, appliquées à un domaine hautement prioritaire pour notre institution et important dans notre société, soit le vieillissement de la population et les actions en faveur du milieu vieillir et idéalement même bien vieillir de la population. La recherche dans ce domaine est transversale, intersectorielle, interfacultaire et même interinstitutionnelle, et elle est très bien arrivée avec plusieurs projets principaux du plan stratégique de l'UDM, notamment la vie repensée et des données avec l'action en santé. Votre leadership en neuroimagerie est très bien établi, comme aussi vos autres approches en sciences cognitives et de transfert de connaissances au bénéfice de la population. De cette façon, votre centre est un modèle de recherche et de formation supérieur et à la croisée des thèmes porteurs pour l'institution et pour tout le Québec. Je conclue in disant, liebe Frau Dr. Friederici, herzlichen Glückwunsch nochmal an Sie in unserer gemeinsamen Muttersprache für diesen Preis und Ihren Beitrag als eine führende Forscherin in den kognitiven Neurowissenschaften. Vielen Dank für Ihren Besuch bei uns im Winter, der dieses Jahr viel zu früh angefangen hat, aber Sie werden ja bald wieder uns entflüchten. Ich bin mir sicher, dass Sie im Rahmen Ihres Besuchs interessante Diskussionen mit unseren Kolleginnen und Kollegen am UGM haben werden und nicht nur hier. Je vous remercie, je finis en disant je vous remercie tous et toutes de votre présence et je vous souhaite un excellent séminaire suivi par vos échanges productifs avec notre lauréate. Merci.
Alors, je vais inviter la lauréate, professeur Friederici, à se joindre. Dr. Pierre Rainville, membre du comité de gestion, représentant le centre. Et je vais remettre le document, le parchemin, ainsi que, non pas en argent comptant, mais un mot qui mentionne le prix de 10 000 $ canadiens, ce n'est pas des euros malheureusement, qui vous sera acheminé, that you will receive very soon. So the parchemin, I'm just going to open it.
Yes, please. This one. There we go. I feel so honored. Thank you very much for sort of providing this, wonderful prize for me. And I think we should, and I should take the prize, also in memory of Justine and Yves Sergent. My talk will be about language in our brain. I recently published a book on this, and in this book, I realized actually that at some point, we have to come to the question of, why we as human beings, have language as a possibility and capability that other animals have not. And I will briefly talk about this today, not only. So, but to give you an entrance into the question I'm following up right now in my mental sort of brain is, how come that children, when they are confronted with language very early on, not only learn single words, but learn to combine the words. So what are you seeing here is the mean length of utterance. And what is compared, it's a case study, 3 cases, a chimp, a hearing child, and a deaf child. And you can see that the chimpanzee, even with the long training they have done with this chimp, was not able to come up with a mean length of utterance that was larger than 1 word. Children, however, they start off by the age of 2.5 years to really, combine words into phrases and sentences. And even the deaf child that has a later entrance due to sign language into the language system, takes off at some point. So what does that mean? I mean, this could mean that generally non-human primates, they can learn words. We know that they can sort of learn words. Dogs can do that, parrots can do that, but they do not combine those into sentences. So there is another study that was published in 2004 that basically gives us the same indication. And they had done larger groups. What they have tried to do is they have compared the learnability of different types of grammars. Take a finite state grammar. This, you can call it a simple grammar, because the rule is after each A element there will be a B element following. And so you can build sequences with that. And it's very interesting that human and cotton-top tamarins can learn those sequences. And learning also means they can detect errors in those sequences. However, when the grammar gets a bit more complicated, the sequences are equally long, but now you have embedded structures, like you can see here in the slide. It's only that humans can learn these. And cotton-top tamarins, even after days of training, are not able to sort of learn those sequences. So, what is the biological basis of the human capacity to process those syntactic hierarchies, those embedded structures? And linguists, they like to draw the underlying structure of the sentences in those trees, and the question is, are we really representing those trees? And how are we managing to sort of process longer sentences? So, the 20th century's view was a view where one was talking about Broca's region. It was called the language motor area, and Wernicke's region, that was the sensory language area. And so this was, what people, discussed a long time, that the one was, the Broca's area was only supporting production and sensory area, only comprehension. Today we know better. We now know that Broca's area not only is supporting production and there sort of the processing of syntax to come up with grammatical sentences, but also comprehension whenever grammar, is responsible for comprehending the sentence. And with respect to the Wernicke's areas, these days we all think rather that this is not only with supporting comprehension, but lexical processing. Okay, so what next? So the next step was that, as I said, that one said, well, Broca's area may be involved in syntax. Nowadays we know it's rather Brodmann area 44, a cytoarchitectonically defined area, and that Brodmann area 45 is rather supporting semantics. I will come up with an example of that later. As I said, Wernicke's area is supporting lexical aspects. We even know more today. We know that these language-related areas are connected by white matter fiber tracks. The dorsal pathways and the ventral pathways. I have sort of color-coded them here for better viewing. And you see that there are two dorsal pathways. The one dorsal pathway is targeting the premotor cortex, and the other one, the purple one, is targeting BA44. So right going into the area that does syntactic processing. So there are ventral pathways, and I will not sort of go into that in much detail these days, but the green one is connecting Brodmann area 45 and 47 to the temporal cortex, and it's supposed to support lexical processing. There is also the Untitled fascicle, that is the red one, and I will come back to that later as well. So, coming from the question I had in the beginning, now the question is sort of, are there cytoarchitectonic or connectivity differences between those areas that make language in the human when we compare this to sort of non-human primates? And for that we need a cross-species comparison. So let's first look at what we have in the literature. So in the literature, what we find with respect to the cytoarchitectonic asymmetry of Broca's area, the following. In humans, there is this asymmetry that we see in adults systematically. It appears to be there in children by the age of 5 for area 45, remember that's the one that does semantics in a simple form set. And 44 only reaches this asymmetry by the age of 11. This is work by Katrin Amos. When you look at the chimp, it's very clear that chimps never show this asymmetry. They always are sort of left and right alike. When you look at the connectivity between the language-relevant areas, this is work by Rilling, what you can see is that there is a very strong dorsal fiber tract, the blue one, in the human, and a very weak ventral fiber tract, the green one, in the humans. When you compare this to the macaque, it's basically the other way around. Sort of the dorsal fiber tract is much weaker than the ventral fiber tract. So these are two things that we may want to keep in mind, and when we take the summary, we could say that there are cytoarchitectonic and connectivity differences in the inferior frontal gyrus, particular Brodmann area 44, and so the connectivity. Louder? Closer? Okay, closer. So the connectivity between the inferior frontal gyrus and the superior middle temporal gyrus, this is also something that differs between the two species. Remember, this is what I told you with respect to behavior when we're comparing the humans and the cotton-top tamarins. When I saw this in 2004 published, I thought, that's interesting. So maybe these two types of grammars are also processed by different areas in the human brain. So we took a very similar experiment to humans and put them in a scanner, trying to figure out sort of, how humans would process these two grammars in their brains. So these were the grammars. And what we did was we had subjects learning those two grammars before they went to the scanner. And in the scanner, then they were presented with correct or incorrect sequences. And we sort of were then trying to figure out which brain areas are activated as a function of those grammars. We had two different groups, I must say. because we were afraid that they would mix up the grammars, during the learning phase. So, interestingly enough, for the finite state grammar, let's call it the simple grammar, what you find is activation in the frontal operculum, but not in Broca's area. When you look at a bit more complex grammar, the phrase structure grammar, you see again activation in the frontal operculum, but now Broca's area, in particular Brodmann area 44, comes in. I should mention that there was no significant difference in the behavior, so they were equally difficult for at least the humans. So with that, I thought here we have an interim conclusion with respect to the following issue. Sanides had already described the frontal operculum as an older, phylogenetically older cortex than Broca's area, the lateral part. And later on, I think I have made this claim, and Katrin Ammons and Calculus as well. So there may be a difference between sort of those two grammars, and also, phylogenetically there may be a difference. At the moment, we can say that at least with the processing of artificial grammar, structural hierarchy in this phrase structure grammar is processed in Broca's area, particularly BA44. But the question is, does this conclusion also hold for natural grammar? German is a wonderful grammar to do that, because they have these nested structures. And so we had a 2x2 design where we had syntactic hierarchy varied in a sense of how many embeddings you have. And also we had to look at the working verbal memory, because the dependencies between those elements also increases as a function, of the embedding. So, for those who know German, here is the wonderful German sentence. Peter wusste, dass Maria, die Hans, der gut aussah, liebte, Johann geküsst hatte. The English translation is, Peter knew that Maria, who knew Hans, who was good looking, loved Johann, kissed. So this is kind of a normal sentence for a German native speaker. And this is one example where we have the same span between the different depending elements, but have other as words in between and phrases in between, not embedded clauses. So in a particular analysis, a region of interest analysis for Broca's area, this is a complex picture, so let me explain. The color-coded green is the Brodmann area 44. The color-coded yellow is the Brodmann area 45. And what you can see is that the main effect of hierarchy, color-coded in orange, is located in the middle of Brodmann area 44. What you also can see is that the main effect of distance, color-coded in light blue, is located in the left inferior frontal sulcus. Those dots and those arrows that go between the different areas mean that in a functional connectivity analysis, we can show that these areas have to work together in order to make the processing of syntactically, sort of hierarchically structured sentence possible. So we can segregate hierarchy from working memory, but they have to work together in order to make this possible. So with that, yes, I could go home and say, well, we know that Broca's area, particularly Brodmann area 44, is supporting syntactic processing. However, what you really want to show is that Brodmann area 44, if the hypothesis is right, increases systematically as a function of increasing the complexity of the sentences. Again, German, a scrambling language, allows to do this very well. So you have here a sentence where you have a subject and two object phrases. Today the grandfather, the boy, the lollipop has given. And what you can do in German, you can sort of put one object in front of the subject. You can even do better. You can put the two objects in front of the subject. So basically, every linguist, although they like to discuss about complexity a lot, all agree that sort of this is an increasing complexity. So what we show here is the following. Here you see that a region of interest analysis in Broca's area. You see the activation for the low complexity sentence, the increased activation for the medium complexity sentences, and even another sort of increased in the signal change for the high complexity sentences. So with that, I think we can be pretty sure that Brodmann area 44 is really supporting the processing of those complex sentences and the syntactic hierarchy increasing sentences. However, meanwhile, linguistics sort of, has developed, and Noam Chomsky has said, well, here is my idea about this. All these complex sentences can be built on the basis of one operation. The operation is merge. So what does merge mean? Merge is an operation that takes two syntactic objects and forms a new one. So what does that mean? Here you have a syntactic object, determiner, one article, the, and here you have a noun, another object, that is ship. And you bind them together, and then you have the ship. And this makes a new object. And then you can add another object, sinks, for example, and then you have already a sentence, the ship sinks. So this is what I have tried to show here on the one side of the slide, what the structure is of those simple operations. So the ship, combines to a determiner phrase, and when you sort of also present then a verb, you have a sentence. Now, looking at where in the brain those constructions are done, what we find is the following. You see here, I have zoomed into the Brodmann area 44, and you see the activation is located, in 44. But I think what is, even more interesting is that the individual peak activity, the blue dots, are all sort of located basically in the same area. So basically, there is very little variation between the subjects in sort of their activation when doing this, performance on merge. So what we can sort of then say, BA44 is involved in building up syntactic hierarchies at all levels. So the very lowest level and also the very complex ones. But how about 45? This is this area, very close anterior to Brodmann area 44. And if it's true that, as some people have argued, that 44 and 45 serve different functions, that is syntax versus semantics, it would be nice if one could show that. So, for example, when you come up with sequences of elements that are supposed to be language-like, and you use non-words like in unflick, like in unhappy, we had sentences where we had sort of the content words were in, and we had the derivations in, like in unhappy. And what you see is that you find activation in the temporal cortex, that is the green one. You have activation in Brodmann area 44, that is the red one, but also strong activation in Brodmann area 45, that is the blue one. However, sort of when you also take out all the function words and all the derivations and just leave only leave the function words and the inflections in, so what you see is that there is only activation, here color-coded in red, in Brodmann area 44, this time color-coded in yellow. So you see that 45 comes in, even when processing Jabberwocky sentences, but with derivational morphology, coding semantic information. That's enough to sort of get this going. And I think this could also be explained, or could explain a number of the sort of different results that we find in the literature, because it's seldom that everybody controls, all these different aspects. 44, however, is activated when only syntactic information is present. So, yes, I have been talking about German. How about other studies in other languages? And there are studies in English, in German, in Hebrew, in Japanese. And what you can see, they have used different types of linguistic constructions, movement, scrambling, nesting, but they all go for more complex sort of sentence types. So you see, yes, Broca's area is activated in all those different languages, but there's also a second sort of cluster of activations in the temporal cortex, the posterior temporal cortex, the temporal gyrus and the temporal sulcus. And so the question that arose at this point is are those two areas always activated together? And if so, are they building a syntactic network? And yes, this is now the hypothesis that we are following up. So the idea is that there is one network that supports hierarchical structures. Broca's area in particular, Brodmann area 44 in the left inferior frontal gyrus, and working together with the posterior temporal cortex. We also had seen that the frontal operculum was active, sort of when we saw this, simple grammar, the artificial grammar. And some people have also argued that the anterior portion of the left superior temporal gyrus is also supporting syntax. So the hypothesis was that there may be two different networks supporting, these different aspects, hierarchical structure and more simple local structures. So here we have the possibility to use diffusion tensor imaging in order to see, whether there are really two different networks. We, as a starting point, again, we took this particular study that I have presented, and we took as the seed point for our calculation of the white matter fiber tracks the individual activation of those subjects in the frontal operculum and Broca's area. So here you see 4 subjects. We have done that for a larger number, and sort of the yellow spot is the activation for this simple grammar adjacent dependencies in the 4 subjects, and the calculation asks, when I see it in this area, where does sort of the fiber track brings me? And you can see that here is the ventral fiber track that sort of is the result of this question. Now, when seeding in BA44, this area is really very close to the frontal operculum. What you see is that there is a totally different network. So the white matter fiber track calculation now brings me dorsally to the temporal cortex in all the subjects that we have tested. So yes, there are different networks supporting the processing of local dependency, adjacent dependency, and hierarchical structure. Yes, so far so good. However, the information from A to B, sort of going through the fiber track via electrical signal, is one thing. However, so the information has to be decoded and encoded at the different spots. And so the idea was maybe we could find that the information transfer within the language network could also be followed up on the basis of the neurotransmitters. And so what you see here as the functional level is the activation from the study that I have presented. And now we do whole brain analysis, and you see, it's the inferior frontal gyrus and the temporal cortex. You see the fiber tracking, and now the question is, how about sort of the information transfer? In order to approach this question, I was working together with Katrin Amunds and Karl Zillis in Jülich in Germany. And what we did was we did a receptor architectonic analysis where we analyzed 14 different neuroreceptors. And we had to sort of now identify the different brain regions that we wanted to look at on the basis of some functional studies, and this is what we used as the interesting areas that we wanted to look at. Remember, this is the study I had been presenting before. So now we have a sort of syntax region that is the inferior frontal sulcus, VA44, and the posterior temporal cortex, that are sort of the red areas. But then we also looked at the different areas that are reported in other studies to support syntax. In order to see whether our analysis would hold really against other areas, we also included other areas and did the same analysis for these. So when you look at the neuroreceptor transmitters, what is usually done, you have a fingerprint. So you look into, so how much is the one or the other neurotransmitter expressed in a particular brain region. And here again, color-coded in red, you see the areas that are involved in language processing, and you have other areas, for example, the V1, the visual area, as the yellow one. And you can already see by the blanket eye that there is a large similarity between sort of those areas that are involved in language. A hierarchical cluster analysis shows this very nicely. Here you see color-coded in red that those regions that are involved in language cluster together by similarity very highly. So I think at this point I could at least make the point that areas that are part of the language network show a highly similar multi-receptor organization. And this appears to be an important prerequisite for the computational processes underlying language functions. As a side step, we did the same analysis for the right hemisphere, and in the right hemisphere, this is not the case. So with that, I think we can at least sort of, make the point at these different aspects, the functional aspect, the structural aspect, and the molecular level. But there are now additional hypotheses I would like to address. The first one is, if the dorsal pathway connecting 44 and posterior temporal cortex is crucial for complex syntax processing, how can we show that this is really the case? Our hypothesis was, if this hypothesis is valid, then the dorsal pathway will not be fully developed in children who still have problems with processing complex sentences. So when would that be? Our colleagues in Leipzig at the Max Planck Institute for Evolutionary Anthropology, they had done a lot of studies in language acquisition, exactly, using sentences that are supposedly complex. They are not long, they are very short. But in German, you can make, those sentences, the dog is sweeping the lion, complex by just, again, putting the object in front of the verb and sort of the subject at the end. And you can see that in a sentence picture matching task where children had to point to the correct picture out of 2, it's only by the age of 7 years and 3 months that they can deal with those object-first sentences. It's only then that they get above chance. So that is pretty late. I mean, all those children can communicate with us, but when you put them in this particular setting where also the verb was a non-verb, not telling you sort of anything about who is doing what to whom, you really had to go for the case marking. That is der versus den. And then, also what we tried to do is to figure out whether they could hear the difference between der and den. Yes, they did. I mean, we have this now confirmed. It was obviously the underlying structure that made the difference. So in a first experiment, we looked at children at the age of 7 and at adults, and you can already see that there is a difference, in the fiber track. So we had sort of at least a hint that, there is a relation. However, for those children, we hadn't done the behavioral experiment at the same time. So now we did, a full study involving 4 different age groups, and children had to sort of go for fMRI, for DTI in the scanner, and we also, did a behavioral experiment. And so we used sentences of, again, varying syntactic complexity, object first and subject first relative clauses. We also had a variation of semantics with a plausible and less plausible content. That is, the big fox is more likely to carry the small beetle than the other way around. But I will now focus here on the syntax effect only and show you the following. Sort of the brains of those different groups really differ with respect to sort of the strengths of the dorsal fiber tract. So you can see color-coded in purple that the fractional anisotropy increases, and that is also testing myelin in sort of particular, increases as the age increases. And this is not the case for the ventral fiber tract color-coded in green. Now, when correlating this with their performance, it was pretty clear that the dorsal information made the right prediction with respect to accuracy and also with respect to speed. So the stronger the fiber tract, so the higher the accuracy, and the stronger the fiber tract, the faster they are. This is not true for any sort of, there is no correlation in the ventral fiber tract. So with that, I think we are at the point to say that the dorsal fiber tract predicts the behavioral performance on syntactically complex sentences during development. Hypothesis 2. If this is true, what I'm trying to convey here, that sort of the strength of the fiber tract, is related to the syntactic abilities, I think then we would predict that the pre-linguistic infant, not knowing grammar, and the macaques, not knowing grammar, should have a weak dorsal fiber tract. So, and in the white matter analysis that is out there in the literature, we already have information, as I presented at the beginning, of those fiber tracts in the human adults and the macaques, with a strong fiber tract in the human adults and a weak one in the macaques. And together with Daniela Perani, we also had the chance to look at newborns. And we found the following, that in newborns, the dorsal fiber tract sort of, is not targeting Broca's area, that is Brodmann area 44, in the newborns. So the ventral fiber tract is sort of already myelized. Myelin is also obviously, bringing forward the dorsal pathway, but only until sort of, or to the targeting the pre-central gyrus. And it's only in the adults that, there is a second stream of the dorsal fiber tract that targets 44. So with that, I think we can say something about the evolution and the development at the same time, and at least with respect to the ability to process syntactic information. Okay, we can sort of leave it there, but we could also sort of take it maybe one step further. Is this a universal thing? Is this also true for other languages? And so we looked at English monolingual brains, at German monolingual brains, and Chinese monolingual brains. And what you see is when you put them together, this is all, you see that there is the dorsal and the ventral fiber tract, so the full network is there. When you look at the different languages, there is some modulation. In the dorsal fiber tract, what you can see is that German, is very strong, a lot of, morphosyntax. English is less strong, the blue one. And the English really, has a stronger fiber tract for the ventral pathway. This could be because, English, is a language that has fixed word order and where you can sort of, do not have to rely all the time on grammatical information, but you can also use semantic relatedness to come up with an interpretation. Chinese, this is not sort of shown here, has a very strong fiber tract also between the two hemispheres. Maybe because it's a tonal language, one has to see. I mean, this is a past hoc interpretation. For English and German, we had a prior prediction that this would be the case. Okay, so I think at this point we can say there is a universality with respect to the language network. It's modulated by the lifelong use of a particular language. And now let me make one last point, which I think is also important. Does language, the network, depend on speech? And so a lot of discussion is that you have to have speech in order to develop these pathways. So we had the possibility, together with colleagues in China, to look at late signers, that is deaf signers that learned sign language late because they were not living in big cities. So we were able to look at late signers and native signers. They learned sign language from beginning on and comparing them to hearing speakers. And you can see that the late deaf signers, color-coded in yellow, are the ones that differ in the function, that is the functional activation of Broca's area from the two others. So deaf native signers use Broca's area as we do as hearing subjects. So then we tried to do the next step, and we had the possibility to look at the white matter fiber tracts of those that were deaf but had learned sign language early, and hearing subjects. And here we look at the dorsal fiber tract, the arcuate fasciculus, color-coded in purple, and you see there is no significant difference between sort of the hearing and the deaf. However, when you look at the frontal assonant tract, which is speech-related, output-related, you see that there is a difference between the deaf and the hearing subjects. So with this, I think we can come to the conclusion that the development of the neural language network is independent of the language modality, sign or sound. It's universal. Okay, here I come to the end. The human ability to process complex syntax is based on a network that involves BA44 in Broca's area and the posterior superior temporal gyrus. These regions are connected by a dorsal fiber tract. This dorsal fiber tract emerges late during development and during evolution. Its strength appears to be crucial for the human capacity of language. So I would not have been able to do all this work without all the wonderful doctoral students I had, the colleagues in my department and also across other departments. And so if you want to read about this, you know, it's all written down in the book, Language in Our Brain. Thank you very much.
Thank you so much for this very interesting demonstration of how you can push questions and questions and questions to answer some of our basic questions about the organization of the brain, in particular for language. I know it's a little bit squeezed, but we may have some time for a couple of comments, questions, if there are from the room here. Any, yep. Let's use the microphone because there's...
Yeah, that's right. You made a comment about the molecular differences between the left and the right hemisphere. Because, and I was wondering whether you have any other evidence about the differences between the left and the right. Because presumably the arcuate fasciculus, the ventral stream, and all these pathways and the architectonic areas also exist in the right hemisphere. Mm-hmm. So I'd like to hear your opinion about what those areas might be doing.
Yes. I know. It's a wonderful question. I have to sort of say that we did take the same areas that we had analyzed in the left hemisphere in the right hemisphere. This was basically because the reviewer wanted us to do that. However, I think you could probably also do it the other way around. You could look at sort of, particular brain areas that are activated during, for example, prosodic processes and see sort of how they are, similar in their receptor architectonic, and then you probably wouldn't see it in the left hemisphere. So I'm not saying that there are no such networks in the left hemisphere. We do. They are there. I mean, also with respect to white matter connections, but they may not involve, exactly the homolog areas.
I have a question about second language acquisition. So, as you probably know, in the second language literature, there are many claims that after the age of 7, and some people say after the age of 4, you are not going to acquire the second language syntax to the same extent. And it seems to me that what you're showing is that you ought to be able to, because the age for syntax, at least the developments that you've shown, are still continuing past the age of 7. So I just wondered if you had some speculation about those kinds of things.
Well, I think this age range, until the age of 7 is very important. I think you could maybe go by, 8, but I think not much further. And so my hunch on this is the following. I think there are certain critical phases, not only with respect to behavior, because this is what we know, but I think there are critical phases in which the brain is plastic and reacts with respect to a particular input. Why am I saying this? I say this because, we try to train children that are not yet at the critical phase of learning sort of, syntax or, showing the fiber track to deal with syntactically complex sentences. So we took 5-year-old children and trained them on complex sentences. First, they didn't learn. Second, there was no difference in the fiber tracking strengths. As a control group, we had children that we trained on words, word learning. They learned like hell, and the ventral fiber tract sort of, increased in the strength. So what I'm saying is there is a critical age, and don't do it too early, don't do it too late. And we know that, when you talk to children below the age of 3 or 4, you already, come up with simpler structures as sort of in communication than with those, kind of sentences I was presenting here. So I totally agree. Do it early and sort of, be simple enough so that the child, can sort of process the sentence.
I think we'll look at the last common question here. Yeah.
Thanks for the beautiful talk. So you showed a lot of differences between groups of subjects, younger infants, older infants, and different types of language, native language, because have you also looked at inter-individual differences within groups, and are there people who are, better at handling those complex German sentences than others, and does that relate to the way that brain is connected?
Well, what we did was after we had done those first experiment with the different subject groups, we had a learning algorithm running over our data, and we had a new cohort coming in, from those 3 different languages, and then we asked sort of the system, can you classify German, English, and Japanese brains? And it works. So yes, you can sort of, do that. But I have to be careful. These are only 3 languages, and I think, it has to also be tested for other languages as well.
Obviously, we could continue for a long time on those topics. I would like, of course, to thank everyone who came here. I'd like to congratulate you once again for a superb trajectory in your work, in your career, and congratulations for this award this year, and thanks for honoring this award by having been second.
Let me thank you again for sort of, having sort of the honor to take the award. Thank you very much.
Yes, Et tout le monde est invité à un café dessert qui est juste en haut des escaliers. Et on aura un peu de temps aussi pour échanger avec le professeur.